Hey IB environmental students, today we're going to be finishing up the rest of those objectives in topic 2 when we're talking about populations, how they change and how we measure them. Let's begin. First we have to review how to define populations, and we're going to talk about how IB expects you to do this. So a population is a group of individuals of the same species found at the same area at the same time. In doing this, we have to be very specific about that space, that area, and we also have to be specific about the type of organism, giving its common name. So, for instance, these are dolphins, but there's a lot of types of dolphins. So IB would expect you to give that type. So, for instance, bottlenose dolphins, and specifically we could pick a location, the Indian River Lagoon. Now, I, for instance, don't know exactly where that is, but either way, we're going to need to be able to do this for some types of organisms, not all types. So let's pick something that we're more familiar with. It's closer to our area. So this is a crab, not just any type of crab. It's got some blue on it, so we actually call it a blue crab, and it is local to our area, the Chesapeake Bay watershed. So we can do this for a lot of types of organisms. You don't need to memorize tons. You just got to know a couple of good case studies. Let's keep going. When we talk about populations, we're going to refer to their different characteristics, and as those characteristics change, we can measure different things. Here's those types of things we might measure and be able to graph eventually. We could look at the size, which is the number of individuals, and that's going to be how we abbreviate individuals for the rest of the podcast. We could also look at the density, which is the amount of those individuals in a certain space or sized area. We can look at the age distribution, which is how many different organisms are at a certain age bracket. For instance, are there a lot of infant-aged organisms versus elderly-aged organisms? And also the dispersion, which is the spatial pattern of those organisms. Let's dig into the spatial pattern first, the dispersion, and we'll hit some of the other ones as we go. So here's common dispersion patterns. You're not necessarily Van Gogh, so maybe draw yourself a box and represent each of the organisms with a circle. You're going to want examples of organisms to do this type of thing. So our first one is clumped. Our example is elephants that we're drawing as circles. These elephants are either in the shade or they are near the tree. They're also in this front area in groups. Maybe there's water in that area, but overall they're in groups, right? They're in these what we're calling clumps, meaning clumped. And they're doing that because that's where the resources are, whether it's shade or food or water. The other option is uniform, which is sometimes found in certain types of bushes. This is an example. Uniform means that they're spatially very strategically placed. They're even throughout the whole area. This is not very common because resources aren't distributed this way. Another example is random. Most weeds, like dandelions, if we look outside, are distributed this way. They're all over the place, and there's no pattern whatsoever to how they are. So these aren't evenly spaced, nor are they in clear groups. So which one's the most common in our environment? Clumped is the most common because it's the most likely how organisms are reacting to their resources. And that's going to get us to the next thing. How are populations limited? Well, populations are limited in their growth of their size due to the fact that there's limited resources. And because of that, this thing called carrying capacity occurs when populations start to grow. Carrying capacity, looking at this graph, is when it starts to level. We'll call that area K. That's the variable we'll use. And it is the number of individuals of a given population that can be sustained infinitely in that specific area. It is specific to the type of organism and the type of area. Why? Because organisms are all affected by different types of limiting factors. And we've learned about a lot of these throughout the year, so I hope you could list out a couple, but here are some. Notice that some of them are abiotic and some of them are biotic, and you would need to know examples of each. All right, let's keep going. Let's talk a little bit more about these graphs. So this graph is showcasing us that populations can't grow forever. But what if they could? What would the graph look like? Is it going to look like the one on the bottom or the one on the top? The one on the top would be if there was no amount of limits. There are no limiting resources. The one on the bottom is very similar to the one we just looked at on the previous slide. So the one on the top is called an exponential growth curve. It is also the shape of a J, and that letter is why we call it a J curve. It is where there is a slow and steady increase in the rate of population growth, but that's going to proceed to start increasing really, really fast all of a sudden. And it's only able to do that because there's no limiting factors. There's no resources that are not in abundance in this scenario. But is that very realistic in the environment? No, it's not. The environment is definitely going to have limits of resources. So most environmental population growths are going to more realistically look like this S shape, called an S curve, or also known as logistic. So here we see that similar exponential growth where resources aren't limited at first, but eventually resources will start to be limited, and that curve will start to slow. And that's different than the top one. And eventually it'll steady out for the most part. And that is what we were calling carrying capacity before. So carrying capacity is not perfect. Sometimes things overgo carrying capacity. Let's talk about what would happen when it overgoes carrying capacity. What about complex carrying capacity? So if for some reason there was so much population growth and the exponential growth went past the carrying capacity, called an overshoot, this is what would happen. Eventually, the organisms would start to consume all their resources, and there would be huge limiting factors. Then their reproduction would begin to slow because there's not enough nutrients for them to be able to have good fertility. Then their deaths would begin to increase, and then we're getting really, really sad, right? We might even go below the carrying capacity at this point, called a crash or a dieback. And eventually we'll fluctuate right around carrying capacity through negative feedback loop or steady state equilibrium. But we do need to understand that carrying capacity is not a fixed thing. It is affected by seasons because there's different abiotic and biotic factors changing by seasons. It'll also be affected by human issues and also natural catastrophes. And also if there's immigration, things coming into the environment, organisms entering, or exiting organisms, immigration. Now we're going to talk about density, and how density can have negative feedback loops within our population growth. The first types of density factors are called independent factors because they are going to affect regardless of the densities of the population. These things tend to be abiotic. They're floods, fires, weather, habitat destruction, or pollution. No matter the size of the population in the area, these things are going to kill off the population in a good bit. It doesn't matter if there was a ton of the population or a little bit about it. All right, so that's compared to these things that are dependent factors that will affect the amount of based on the amount of individuals in the area. So the denser the population, the more the effect usually. So here's some external factors, external to the organism. These, you'll notice, are very much biotic examples. Competition is going to increase if there's a higher density because there's going to be less resources available for each individual. We can think about diseases as a really good example. Epidemiology is the study of the spread of diseases, and we know that if there's a dense population, it's more likely you're going to bump into somebody and a contagion can occur. This is why we're worried about Ebola. It's more likely you're going to get coughed on if there's a denser population, and diseases would spread. There's also internal factors that are within the organism, such as certain organisms stop their ability to be fertile or breed if the density is too high and there's organisms too close by. That's, for instance, possible with paramecium. That's very common for single-celled organisms. And they'll see a very slow, and it'll hit carrying capacity on its own just because the population is too dense. So that's the effect of density on a population causing negative feedback loops. What about other things? Well, we can also look at predation overall as a negative feedback loop. We've looked at this before, and here's our case study. The link's in the hair. You do need to know this case study, and so I would write it down, and I might even draw a simplistic version of this cycle. So we can see that over time, these things are at a steady state equilibrium. They fluctuate, but overall, they're really steady at a carrying capacity. It's not going too far over. We just see overshoots and diebacks occurring very frequently. We can also talk about different fertility strategies, and we call those R and K strategists. And we're going to talk about two different examples. The first example is the R strategist, also known as R-selected species. These species reproduce very early in their life, and they produce many organisms because few of those organisms are going to survive. They like to live like rock stars. Another type is the K-selected. They reproduce pretty late in their lifetime. They reproduce not very many organisms, and they really like to parent them because it's all about the kids. Oh, the kids. So which of these two do you think we're more like as humans? The K-selected, think about it. We reproduce pretty late in our lifetimes. We don't reproduce at age 5. We reproduce once we hit puberty and hopefully even later, right? And we also will take care of our species, our young, for at least 18 years is the common thing in most places of the world these days. Things like rats and insects and mosquitoes and other sorts of weeds, they are selected. They like to make lots of babies. So on our next slide, we're going to see a chart that kind of goes through these examples, and I'm going to go through it quick because you're going to get to practice it in class. You can also peek at this graph, and you can see that the R selected ones are on the high exponential part of the graph, where things are reproducing fast, versus the K or near carrying capacity. They don't reproduce very much, and that should make sense. So here's our graphic. Pause and write them down and see this comparison of the different things for K and R strategists in more detail. And pause to get more of these down. Do notice which are more common in different parts of succession. This should make sense based on how much they're reproducing and how much they like stable versus unstable conditions. All right, let's keep going. Related to the R and K strategists is survivalship curves. Survivalship curves are described as the life expectancies for different species. You'll see that this graphic shows age of the species over percent surviving. The late loss curve is the red one here with the rhino, and that's mostly going to be K-selected species just like ourselves, where we usually, most humans will live for quite a long life, and then once we get old, we start to kind of die past reproductive age. Versus early lost species like rats and fish and mosquitoes, it's very unlikely they will live will make it past infancy, and then if they do make it past infancy, which is wonderful, they do tend to live pretty well, but a lot of them die really early in their lifetime. And then the last one is the constant loss curve, which refers to an intermediate and steady mortality. Think squirrels or deer, which could get hit by a car at any time, and there's so many of them, and they're just BAM! Dead. It could be any time in their life they get hit by a car. So that's what this constant loss one is. So it's clear K, R, and then there's some in the middle. There's a lot kind of all over the place here, but it's clear that the K strategists and the R strategists are at the extremes. It is important to now look at the human impacts on natural populations. We can see some of these things. They're kind of really sad. I'll highlight a couple. But overall, humans are destroying habitats, sadly. They add pests and disease to the populations. Sometimes they get rid of predators. Think about the wolves are gone, and then the deer are reproducing like crazy. We also add invasive species that can kill off the natural ones. We over-harvest things like trees, and because of all this, the chemical cycling and the energy flow within our system is all wackadoodle. So why might we measure then our population? Well, we want to see change over time for succession reasons, but we also might want to see the impact of human issues like we just described, so we could try to solve them. So let's say there's a toxin from a mining or a landfill, or maybe there's an oil spill. So for instance, here's an example of the Exxon Valdez oil spill that happened in the late 80s, and scientists wanted to track how the populations of the species in that area, mostly in the Alaska area, were doing over time, to see if they could help them. We could also think about how we might measure these effects or these changes. We could do Lincoln Index to get population size. We could do quadrats over time. We could also use a cool new technique like satellite imagery to see if this amount of different populations is changing over time. This is the Amazon rainforest, and we can see deforestation between 2000 and 2006. How would we do this? We could do a pre and a post of the area. We could also compare an area that got impacted versus an area that wasn't ever impacted. Last but not least, what is the legislation that goes with this? Well, we call it an environmental impact assessment. And it is prepared before a developmental project or something that's related to land use changes. For instance, if we convert a forest or to an agricultural field or a golf course, we would do this. So think about how much forest was in Fairfax County. This is Herndon, and look how much development has occurred. So the big idea is we want baseline data about the population size so that eventually we can see what is the human impact on the abiotic and the biotic community, so we can make sure that we as humans humans are taking care of the environment. Good job, you guys. You made it. Whew.